Someone left their mic on.
Yeah, I just got it. I've been impressed that that's the first time that's happened this semester. I have meetings with professors and all kinds of highly paid people that can't comprehend that we don't want to hear them typing on their computer for an hour-long meeting. There was somebody, you could hear them chewing and eating their lunch. It was... Okay. Okay. So you got a full screen slide now?
Yes.
Okay. So the first week we talked about the body, and in that lecture we learned about the levels of organization. And at the very smallest levels of organization, we had the chemistry level, which had atoms and molecules, which we talked about last week. This week we move up one level, and we're going to talk about cells. And the next week we'll move up another level and talk about tissues. And remember, tissues are a collection of similar cells. And so today we're going to learn about how each individual cell works. I'm guessing some of this you've had before. You've probably taken some basic biology, and that's, I'm sure, is something that was included there. But remember that all living things are made out of cells. Some living things are one cell, like a bacterium. Other things are many, many cells. We're made of trillions of cells. And when you look at a cell, there are a lot of parts there, and we're going to talk about just a tiny fraction of them. But we can break it down into three fundamental parts or portions. First, we have the plasma membrane, which we also call the cell membrane. It's the same exact thing. That makes the outside of our cell. We talked last week about what that was made out of. Do you remember what the cell or plasma membrane was made out of? What kind of molecule?
Phospholipids?
Yes, phospholipids. We're actually going to talk more about that and kind of revisit that in a little bit. But the plasma membrane, the cell membrane, is made of phospholipids. And I remember somebody made a very good conclusion or explanation saying that the phospholipids help to differentiate the inside of the cell from the outside of the cell. As animals, we don't have cell walls. Plants have cell walls in addition to the plasma membrane. Because we don't have that wall, the plasma membrane says where one cell stops and the outside of the cell starts, or a second cell starts if they're right next to each other. On the outside of that cell, we have extracellular fluid, which we like to abbreviate ECF. That is any sort of fluid. It's usually going to be like a salt water type solution that is out here. How much of that there is depends on how densely packed the cells are. If you have some sort of tissue where the cells are packed in very, very tight, there's not going to be very much extracellular fluid. But in other parts of our body, the cells are loosely packed. We'll see that next week. And if the cells are loosely packed, something has to take up that space. And that's going to be extracellular fluid. On the inside of the cell, there are lots and lots and lots of fancy parts. We're going to learn about some of those fancy parts today. But as a whole, everything inside of that cell is called the cytoplasm. That includes these organelles, which we'll learn about, which are the functional parts inside of a cell. But it also includes this fluid that is inside of the cell. And we'll learn the term for that fluid later on. But you can think of that as intracellular fluid, or ICF. So here's the same diagram blown up. And we can see the most prominent thing here is the nucleus, right? And we don't know what the nucleus does yet, but it is very, very big and prominent. In most of your cells, if you looked at them under the microscope, the nucleus may take up a third to a half of the cell, and most of these other things you wouldn't even be able to see. This is not drawn necessarily to scale. The mitochondria are not this big. The ER is not that visible. What you're going to see under the microscope is a nucleus, and it's going to be a purple dot. When we start looking at slides next week in lecture and for the lab, in the tissues lecture, we'll see what these look like under the microscope, and believe me, you are not going to see any of this other stuff. So like we said, the plasma membrane or the cell membrane is primarily made up of phospholipids, but it is not completely made out of phospholipids. Although most of the molecules in the plasma membrane are phospholipids, there is some cholesterol. Now, cholesterol we talked about last week as being the fundamental steroid, the molecule that all other steroids are made off of, but cholesterol itself serves some functions. One of them is that it buries itself in the plasma membrane and causes the plasma membrane to be a little bit more fluid. It helps it to kind of float around. Those phospholipids don't stay in place. When we see a diagram coming up of that, keep in mind that these things float around. It's like a raft on water. It can float around. And cholesterol keeps everything from seizing up like glue. So this was that phospholipid. And remember, it had two primary parts. We had the head and the tails. And in the head was the phosphate portion. This is the hydrophilic part, the part that likes water. And then the tails are the hydrophobic part, the part that does not like water. And these are made out of fatty acids. When we take a whole bunch of phospholipids and put them in water, they will naturally form this phospholipid bilayer. So they make two layers, and they orient themselves so that the tails point towards each other. Remember, imagine that everything else on this slide is water. Those tails are hydrophobic. They fear water. They want to get away from the water, but they like other hydrophobic things, which is the tail on the other layer. And so they find a friend, and they just huddle in the middle here, while the hydrophilic heads touch the water on the inside of the cell and the outside of the cell. Now here we're looking at an actual plasma membrane. It's not just this basic lipid bilayer diagram, but here we're looking at a diagram of a cell membrane. And we can see that there is a lot of stuff here. We see hundreds and hundreds of the phospholipids, but there's plenty of other things. These kind of honeycomb yellow things are the cholesterol. And then we have all these very big things in here. Almost all of these are proteins. Remember, proteins were made up of amino acids, and a lot of times they have a function. Either they're going to be an enzyme that carries out a reaction, or they're going to be structural, or they're going to serve some other purpose. We're going to talk a lot about these. I'm not going to go through everything that's in this diagram, so we're going to talk about them later. But I think on another slide, we're going to mention that some of these proteins serve the function of acting kind of as a marker or identification of what type of cell this is. And that is the proteins that have these little sugars coming off. They get these little tails or fingers sticking up. Those are carbohydrates or sugars that are usually attached to proteins. So here we see there's this blue thing coming off this blue blob. There's a blue thing coming off that blue blob. That is a sugar, and different cells are going to have different sugars on the outside acting as flags to tell other cells what kind of cell they're looking at. These glycolipids are the same idea, except instead of being attached to a protein, the sugar, the glyco part, is attached to a lipid. Now that membrane, we said at its most basic function, differentiates and separates the inside and the outside of the cell. But there are a lot more complicated functions of that cell membrane. One is it contains receptors. And if we go back, I think there's got to be a receptor on here. There isn't. So a receptor, imagine this blob right here. If you can see my cursor, you can see my cursor, right? Somebody?
Yes.
Okay, thank you. Imagine this blob is a receptor protein. A receptor protein is going to sit there on the surface waiting for something to come along, float in, and stick to it. And that's going to help that cell tell what's going on around itself. If it sees certain types of chemicals that are binding to those receptors, it knows maybe I should try to grow, maybe I should stop growing, maybe I should try to leave, maybe I should store up a whole bunch of ATP because something's going to happen. Once that receptor becomes activated, a lot of times it will activate other proteins in the plasma membrane, and those would be second messenger systems. Cell signaling, how it talks amongst itself inside of a cell, is a very complex process we're not going to talk about, but there are usually dozens and dozens of proteins involved. It's protein A activates protein B, which activates C, D, E, F, G, and H, and so on and so on. That would be the second messenger system. Some of those proteins are also located in the plasma membrane. We also have enzymes that were buried in the plasma membrane. And remember from last week, enzymes are the proteins that carry out reactions. They're catalysts. They make things happen, and they make them happen very, very quickly. Next we have channels. If we go back, a channel, this here we have one labeled, and notice it's just a pathway through the membrane. And we'll see how that works later, but that is literally what it is. It's going to allow some things to pass through the membrane that wouldn't normally be able to pass through if that hole wasn't there. Carriers are very, very similar to channels, except they usually are going to be more specific. They're going to only allow one particular thing through, and sometimes they are pumps. Rather than just allowing things to float through, they are actually going to act as pumps and use energy along the way, and we'll talk more about pumps later on. I forgot to mention about the difference between an open and gated channel. This channel here is open. Notice that there is free flow going through that channel. A gated channel would have a little flap kind of in the middle here that would allow the channel to be open. Imagine the flap is down, allowing things to flow through it, or it can pop up and block things going through that channel. So that would be a gated channel. We also have the cell identity markers, which are what I was talking about with the little sugar flags sticking up off the top of the cell. And then finally, there are cell adhesion molecules. In most parts of our body, the cells are right next to each other. And not only are they right next to each other, but they are stuck to each other. And that's usually going to be carried out by proteins. You can have protein sticking out of one cell and a protein sticking out of another cell, and when they touch, they lock together, and so the two cells that own those proteins are now glued together. So those are cell adhesion molecules. Questions about the plasma membrane before we move inside of the cell? Anything? Okay. So we have a very long list of cell structures here. We are going to talk about, I think I went through this earlier. I believe there is only one of them that we are not going to talk about. And that is a vesicle. Okay? A vesicle is a storage place in the cell. Under the microscope, it looks like a little white bubble, and it is literally like a little storage. It's like a closet or something in the cell where cells can just put stuff in to hold it and wait for it later. I think everything else on this list we're going to specifically talk about in the lecture. All of these things are also on the cell lab. The lab gives you a table and has all of these things, and you have to fill in the function of each thing as you go. So the first thing we talk about are ribosomes. Now, ribosomes are very important. They're more complicated than this figure makes it look, but you don't need to worry about all the little details. So a ribosome, its function is to make protein. If you can remember that it makes protein, for right now, that'll be good enough. At the end of the lecture, we'll talk about that process a little bit more. We'll talk about this mRNA and what it's doing here. But right now, you don't know what mRNA is. So right now, ribosomes are an organelle that makes protein. And an organelle, like I mentioned earlier, is a little functional part of the cell that carries out one little job to help the cell as a whole function and stay alive. Next, we have the endoplasmic reticulum, and we abbreviate endoplasmic reticulum ER. And the ER comes in two flavors. We have the rough ER and the smooth ER. This, I like the person that named this because it makes sense. You can see it both in the diagram, the drawing, and also the microscope picture, particularly on the right. The rough ER has these little dots on them. And you can see it, like I said, in the diagram, and also over here on the microscope slide. And those dots are actually ribosomes. Those ribosomes are on the surface of that endoplasmic reticulum, making protein like ribosomes do, but as it makes the protein, it actually injects the new protein inside of this endoplasmic reticulum. And the endoplasmic reticulum is made out of phospholipids, just like the plasma membrane. Imagine taking some plasma membrane and kind of folding it up into a whole bunch of layers and putting it inside of the cell. That is the ER. But there's open area between those folds. That is where the proteins being made by those ribosomes are being injected into. And so the rough ER is the site of protein synthesis. Some ribosomes are not stuck onto the ER. Some of them just float around, and they make proteins also. So not all proteins are made in the rough ER, but basically the only thing that happens in the rough ER is where some of the proteins are made. Now you can see, particularly in the diagram here, that the ER continues further out from the nucleus. The rough ER is right next to the nucleus. As it continues out, away from it, the ribosomes stop. This is where the smooth ER is. It's the same structure as the rough ER, but since there's no little dots on it, we call it the smooth ER and not the rough ER. Now, remember the dots were ribosomes, which made the protein. And so if there's no ribosomes on the smooth ER, we can't be making protein there. The smooth ER is one of the primary locations where lipids are made. So the primary function of the smooth ER is it is where the lipids are made. Next we have the Golgi. This has a lot of slightly different names. I called it the Golgi. That's kind of short for Golgi apparatus. Some places you'll see it called the Golgi complex. Like the endoplasmic reticulum, it is made out of plasma membrane folds, but notice in this diagram, it does not touch the endoplasmic reticulum. So the rough ER and the smooth ER are continuous. They are one thing. They're connected. The Golgi is very, very similar, but it is not actually connected. It sits right next to it, and its primary function is to take the proteins and the lipids that are made in the ER, receive them, kind of package them up, and then send them wherever they need to go. In some situations, that might be a different part of the cell. In other situations, that might be outside of the cell. So think of the Golgi as kind of a shipping warehouse. It receives things from the ER, packages them up, and then sends them out wherever they need to go. Next here are the mitochondria. So mitochondria, the word you're looking at here, is plural. The singular form of that is mitochondrion with an O-N on the end instead of an A. This, the kind of cliche term we always use, it is the powerhouse or the power plant of the cell. I think we learned last week that ATP was made in the mitochondria. If we didn't, we certainly learned about ATP. And the ATP is made in the mitochondria. There is no other place in the cell where ATP is made. Now, this is a very interesting little organelle. We're not going to talk too much about the detail, but it actually has 2 layers of phospholipid. And when I say 2 layers, it's 2 phospholipid bilayers. So actually 4 layers of phospholipid. Does anybody who took basic biology, do you remember why, or what the theory is why mitochondria have 2 layers of phospholipids? Nobody? So the idea is that the mitochondria actually used to be bacteria. And that long time ago, some animal cell swallowed it up thinking it was going to eat it, and then changed its mind and said, well, this bacteria is making a lot of ATP. If I don't kill this bacteria, I might as well let it live inside of me. I can take the ATP that it makes and use it for myself. Because it's better at making ATP than I am, so I'll just let it do its thing. I'll just let it live and take its ATP. Nowadays, it looks nothing like a bacterium. It is just a structure inside of our cell. But if you look at it, you can see the 2 membranes. Now imagine on the outside is a layer of membrane that was originally part of some cell millions and millions of years ago that wrapped around this bacterium. And then inside are the remnants of the old bacterial membrane with these folds. And those folds are where the ATP are made. Next we have peroxisomes and lysosomes. These look very similar. There's no way that you would ever look at a microscope slide, or really even a diagram, and say this one is a peroxisome, this one is a lysosome. Usually lysosomes are bigger than peroxisomes, but you're looking at the slide right here, and whoever drew this, made the peroxisome bigger. And so you can't go by that. That's not a question that I could ask you at all. I could ask you what the function of them are, though. So a peroxisome is primarily for breaking down lipids. That is the primary function of a peroxisome. It has peroxides in it, think hydrogen peroxide, and it uses that to break down lipids. A lysosome, like I said, is normally bigger than a peroxisome, and it has enzymes, proteins, that break down things. So a peroxisome has peroxides, small chemicals. Lysosomes have big enzyme proteins, and that is because a lysosome is designed to break down cell structures. Other parts of the cell, like the mitochondria and the Golgi, they get old, they get worn out. And so we need to replace them, but what we can do is we can actually recycle their parts. And so a lysosome will actually swallow up a mitochondria, break it down into its basic parts, put those parts back out into the cell, and then the cell can use them to make a new mitochondrion. So peroxisomes break down lipids, lysosomes break down large cell structures, but they both are breaking things down. Next is the cytoskeleton. The cytoskeleton is like the tent poles holding the cell membrane or the plasma membrane out. When we look at a cell diagram on a slide or in a book, it is two-dimensional. Cells are not two-dimensional. If you look at them in a petri dish, they're going to be more or less flat. They're going to be a little thick, but they're not going to be very thick. However, in a three-dimensional organism, most cells are going to be they're going to be round, or they're going to be cube-shaped. They're going to be very tall and wide. They're not going to be flat. We'll talk about, there are some flat cells, and we'll learn about shapes next week. But most cells are going to be big and round. And so we have these poles, the framework holding the cell and giving it its shape. We have three types of those poles. We have microtubules, microfilaments, and intermediate filaments. There's there's two primary differences between these. The size and what they're made out of. And so I need you to know which one is the biggest, which one is the medium, which one is the smallest, and I need you to know what each one is made out of. I don't need you to know the 25 nanometers, the 7, and the 8 to 12. I don't care about how big they are. I need you to be able to rank them for me. So the microtubules are the biggest one. They are made out of a protein called tubulin. Tubulin makes up each one of these little balls here that we see in the diagram, and then they kind of come together and they make a little helix shape. It's very, very tightly packed. These are really strong. We'll learn more about the function of these later on. They not only play a role in holding the cell shape, but they play a very, very important role in cell division. We also have actin that makes up the microfilaments. So these are the smallest ones. Microfilaments are the smallest, and they are made out of actin protein. Notice this isn't nearly as tightly wound as the tubulin is over here. You have like two strings and you just kind of wrap them around each other. Then we have the intermediate filaments. Intermediate means middle, right? So that's easy to remember. The intermediate filaments are the medium-sized ones, and they are made out of keratin. This is kind of a mixture of the structure of the other two. We have a whole bunch of strands, but it's twisted together like the microfilaments. Then, I think this is the last one. No, we got a couple more. So then we get to the nucleus. Now, the nucleus plays a lot of different roles in the cell. A lot of them we're not going to talk about today. The easiest way to think of this is, like the slide says, is the control center of the cell. Everything else that happens in the cell happens because of what happens in the nucleus. I want you to think of the primary function of the nucleus as to contain the DNA. Now, it does a lot more than that, but the DNA, we can't live without the DNA, and it is stored in the nucleus. DNA never ever leaves the nucleus other than during cell division, which we'll see. But the nucleus has a double lipid bilayer again. Just like the mitochondria did, there are two layers here. We call that the membrane around the nucleus the nuclear envelope, or the nuclear membrane. Same thing. Some people call them different things. And then embedded in that membrane are pores. Now these are proteins, just like the channel proteins on the plasma membrane. They're going to allow things to get in and out of the nucleus. Some things are going to have to get in, other things are going to have to get out. Inside the nucleus, we can see a couple of things. The little squiggly lines are the DNA. It is labeled chromatin. You don't know what that is yet, but we'll learn what chromatin is later on. But it is just DNA. And then in the very, very center of the nucleus, there's a dark dot called the nucleolus. This nucleus only has one nucleolus or nucleolus. That is not always the case. You can have 2, 3, 4, you can have a dozen of them. In that case, there would be nucleoli. So Li on the end rather than US. Ribosomes are made in the nucleolus. Remember, the ribosomes make the protein. That's the dots, the rough part of the rough ER. They make proteins, but the ribosomes themselves are made in the nucleolus, inside of the nucleus, meaning they then have to leave and come out these nuclear pores. Now we can start looking at what we call appendages. So these are not actual appendages like arms and legs, but they look like it, so we call them that. And we have three main types. We have microvilli, cilia, and flagella. Structurally, they are very different from each other, and functionally, they have very different purposes from each other. They do not at all do the same thing, even if they might look similar. So we'll start with the microvilli. These are really, really small. Notice that here we have zoomed in on these cells. So what you're looking at, this is actually the fold of an intestine. On the inside of our intestines, we have lots of little folds to increase the surface area because our intestines absorb food. And so the intestines are themselves actually have little folds. And so we're looking at one of those folds, and on the edge of that fold, so the food would be out here, we have these cells. And if we zoom in on those cells, the cells have these little hairs on them. Now all they are is little folds in the plasma membrane. So I'm going to take the plasma membrane and just do this with it, and you get these little hairs. That's all they are. They don't move, they can't do anything. Just like we put folds in the intestine to increase surface area, we put folds on the plasma membrane of the cells in the intestine to even further increase the surface area. The more surface area we have, the more food our intestines are touching at any given time, so we can absorb more nutrients. Bigger than microvilli are cilia. Now when you're looking at these diagrams, they look very similar, but structurally they are very, very different. Cilia are bigger than microvilli, and where microvilli are just folds in plasma membrane that can't move, cilia actually move. It's a fold in the plasma membrane that has a microtubule or part of the cytoskeleton stuck up in it. And when you have a tent pole in something, if you move the tent pole, you can move the fold or the tent with it. And so cilia are designed to move water around a cell. And so one way or one place that we have this is in our airways. We have mucus in our airways, and that mucus needs to move. It sits there, and its purpose is to catch the junk that we breathe in, but we don't want mucus just sitting there for days and months and years because it's going to get really nasty and we need to get rid of it. So the cilia actually will beat. It's like the finger going like this, And as they move, they create water currents, and the water currents are actually going to move the mucus. So cilia can move, whereas microvilli can't. Flagella are basically very, very large cilia. But rather than beating like this, they actually make a corkscrew movement. And there's only one human cell that has a flagella, and that is a sperm cell. There are no other flagella in humans. So here's a question. Where does protein synthesis occur? And the key word here is where, not what carries out protein synthesis, but where does protein synthesis occur? Who thinks they know? Who thinks they know? Yes. So we got a couple answers for C, the rough endoplasmic reticulum. What makes it the rough endoplasmic reticulum? Why is it rough? Yeah. We have ribosomes on the endoplasmic reticulum making it rough. And what do ribosomes do? They make protein. And so ribosomes make protein, but a lot of our protein is made in or on the rough ER. Okay. What time is it? I forgot to bring my phone in. So how many slides? Let's do it. I'm debating when to take a break. Let's keep going. Okay. So the next thing we need to talk about is the transport of things in and out. Actually, never mind. Let's take a break. 5-minute break. We'll take two. We'll take 2 breaks today. So 5-minute break. So my clock says 6:49. We'll come back at 6:55. So did anybody do anything fun this weekend? Nobody did anything fun.
Working.
Hey, considering what some people are going through, working is not something to complain about right now, I don't think.
Not at all.
You can't see it, but there's a deer out here.
Are you going fishing today?
That's my hope. It looks like we shouldn't have any rain. And so we'll see what time we finish, but it doesn't get dark until almost 9 now.
Well, I hope you get to go.
I probably will. It's just the mosquitoes get really, right about 8:45, 15 minutes before it gets really dark, the mosquitoes just get really, really bad.
Oh, yeah.
Luckily, they're not bad during the day. Considering we live next to a kind of a swampy lake, it could be bad all the time, but it's not. They only come out when it gets dark. So,
For my puppy.
ooh.
Okay, wanna go?
Let's,
Okay, go on.
dog's not going to help you cheat on the exam, is he?
I don't think so. She's not that smart.
I hope she didn't hear you call her dumb. Okay, so let's pick up where we just left off. And so we were going to talk about the transport or the movement of things inside, in and out of a cell. This is really, really important. Cells need to get into cells, or nutrients need to get into cells. Cells themselves don't get their own nutrients. They have to have nutrients supplied to them. And we have other things that need to get out of a cell. When we take something in, we take nutrients in, and we convert them, we use them, we get waste products. And we need to get those waste products outside of the cell. We also need to control how much water is inside of our cell. If the cell starts to dry out, we need to get more water in. If the cell starts swelling up, we need to get water out. And so we need to very tightly control what gets in and gets out of the cell. We have two primary types of transport. We have passive transport and active transport. Active transport requires ATP, so it requires energy. Passive transport is not going to require energy. For the most part, passive transport is simply going to allow nature to do, or Mother Nature, the physics and chemistry to do what it wants to do. Active transport is going to force chemistry and physics to do the opposite of what they want to do. Think about having water on a hill. You can passively allow that water to drain down the hill. But if you want to go against gravity, you're going to have to use energy to pump it back up to the top of the hill. So the most basic type of passive transport is diffusion. And you are completely aware of diffusion whether you know it or not. Imagine, well, I just realized the metaphor that the example that I always use doesn't work when we're not in the same classroom. But what I normally say is imagine there's someone in the back of the classroom that puts on some very, very cheap, smelly cologne. And you're in the front of the class. They put it on themselves in the back of the class, but eventually you are going to smell it in the front of the class. You can smell it in the front of the class because of diffusion. Diffusion just means things move from where there's a lot of something into where there is little of it. So in that example, when that person puts the cologne on in the back of the class, there's a lot of cologne in the back of the class. and it is slowly going to spread out. Think of taking a glass of water and putting a single drop of food coloring in it. Initially, the food coloring is right where you dropped it, but eventually that food coloring spreads out through the entire glass. It's a very big glass. And the whole thing turns colored. That's because the food coloring is diffusing through the water and spreading out equally through the whole thing. But with a cell, we have a plasma membrane separating the inside and the outside of the cell. So imagine in this situation, we have something on the outside of the cell, a whole bunch of it. Let's say it's sugar. Let's say it's sugar on the outside of the cell, and we have no sugar on the inside of the cell. Basic physics, diffusion, says that this sugar wants to diffuse into the cell. But we have a plasma membrane in the way. So it can't just very quickly move over there. Sugar, this molecule that's here, is something that can get through the plasma membrane somewhat. It can't just go through it like it's not there, but over time it will slowly equal out. And so that we have the same amount on the outside as we have on the inside. That is diffusion. Some molecules, though, can't get through the plasma membrane at all, which is why we're going to have some of the other types of transport that we'll talk about in a few slides. Now, very similar to diffusion is osmosis. The basic principles are the same. It's passive, but here osmosis applies only to water. Osmosis is the movement of water through what we call a semi-permeable membrane. Now, semi-permeable membrane is a term that gets used every time someone talks about osmosis, and never any time someone is not talking about osmosis. A semi-permeable membrane is a membrane that allows some things through, but not other things. So imagine we're looking at this beaker here, and we put this membrane down the middle. Now the membrane goes all the way down to the bottom, so we can pour one solution on one side, one solution on the other side, and they're not able to mix unless they go through the membrane. Now what we've poured in here is two different sugar solutions. On the left, actually this is probably your left. On your left, we have a low concentration of sugar solution. On the right, we have a high concentration of sugar. Now, based on diffusion, the sugar would want to move from the right over to the left. But sugar is not able to get through our membrane. It just can't. No matter how much it wants, it can't get through. We have the same amount of water on both sides. And so, in the absence of sugar, if there was no sugar involved here, there would be no reason for water to move from one side to the other. But you can see in the diagram, water moved from the left over to the right. In osmosis, the water moves towards the high concentration of solute. Last week we talked about solutions. We said a solution was made up of a solute dissolved in a solvent. So in this situation, we have the sugar solute dissolved in the water solvent. And the water is moving from the left over to the right. It's going to where the high concentration of sugar is. Its goal is to try to equalize the sugar concentrations. Now notice, as we move the water from the left to the right, on the left, the sugar got more concentrated. The sugar molecules are packed in tighter, and as it moved over to the right, the sugar molecules on the right actually got more diluted. They're further away from each other, And now they are roughly the same concentration. We have more sugar solution on the right than on the left, but the two solutions are the same concentration. Does osmosis make sense to you? Osmosis is an important concept.
Yes.
Okay. Just remember, in osmosis, water moves towards the high concentration of solute. And in this class, that solute is either going to be sugar or it's going to be salt. And we'll see the salt working in osmosis in just a little bit. Another thing that can happen is filtration. This is passive. We are not directly using energy. You are familiar with filtration. If you put water on top of a filter and let it flow through that filter, gravity is going to pull it through. The filter allows some things through, but not other things, right? And so here we have a blood vessel, and it is going to filter things leaving that blood vessel. We want plasma and very small nutrients like sugar and oxygen and things like that to be able to leave the bloodstream, to get to the cells that are over here. But we don't want big things like blood cells to leak out. So this is filtration. We have blood pressure that is pushing water on the wall of this blood vessel. And that wall of that blood vessel is actually a cell. So we are using a single cell. This is the nucleus. That black dot is the nucleus. We're pushing water through that cell. The water, the salt, the sugar can filter through that cell, but there's no way a red blood cell can pass through another cell. So filtration is pushing things through something using pressure. Here we see that zoomed in more. So we have stuff in our blood. We have large molecules, we have small molecules, we have water. There's blood pressure in there, and that blood pressure pushes the small molecules and the water out, where the large molecules stay in the blood vessel. Now, before, we talked about diffusion. Diffusion was just things moving from high concentration to low concentration, where there was a lot of something moving to where there was a little bit of something. And in that example, on that slide, that sugar we were talking about could move through the plasma membrane. It could diffuse right through it. It may slow it down a little bit, but given enough time, it can move through. At that point in time, though, I said some things can't move through the plasma membrane at all. And so, to allow them to diffuse, we have to have facilitated diffusion. This is in contrast to passive diffusion. This is facilitated diffusion. But confusingly enough, this is still a passive transport process. There is no energy being involved here. We simply facilitate physics to do its thing by giving it a hole in the membrane to go through. This is a channel protein like we saw before, and the molecules can move from the outside, where there's a whole bunch of them, through the channel protein to get to the inside. The carrier protein on the bottom does the same thing, but it is selective. Notice here, it is only allowing the oval molecules through. The channel up top, while we can't see any ovals going through it currently, there's already an oval on the inside. So the channel is non-selective. Anything that can fit through it will fit through it. But the carrier protein will only allow certain molecules to cross. But neither one of them uses energy. There's simply a hole or a pore that allows diffusion to happen because it can't get through the plasma membrane itself. And then finally, we have a pump. Pumps are active transport. Pumps use energy. In the real world, in real life, when you have a pump, either you're going to have to plug it into the wall or you're going to have to pump it yourself, right? You have to put energy into this. And we are trying to do something that doesn't want to normally happen. if you want to put air into a tire, if you have a tire that's already pumped up, the air wants to come out of the tire. But if you want to put more in, that's not the direction it wants to go. And so you're going to have to use energy to make it happen. Now there is a lot of detail on this slide. We're going to come back to this pump in either the neuron or the muscle lecture at the end. I think that's middle and the end of the semester. We'll come back to this one. This is a very important one. This is the sodium-potassium pump. This pumps sodium out of the cell and potassium into the cell. But normally, there is a lot of sodium outside the cell and very little in. And so the sodium wants to come in, but we're trying to get it out. And so we have to use the pump. And usually, we have a lot of potassium inside the cell and very little potassium out. Yet we want to take what little is still outside and get it in. So again, we need energy. This pump does both at the same time. It uses ATP as a source of energy, pumps 3 sodiums out and 2 potassiums in every time you make that pump go. So it is going against what physics wants to have happen. Another type of active transport is endocytosis. So endocytosis is the process of bringing things into the cell using part of the plasma membrane. So in the diagram, when we look at this, we see the yellow line here is the plasma membrane. And we are actually going to use that plasma membrane and fold it around something and to make a bubble. That bubble pinches off and comes into the cell, and the plasma membrane reseals itself back behind it. So here, we are actually swallowing a bacterium. This is a whole bacterium. Imagine this is probably a white blood cell. And it is going along. It is grabbing that bacteria, swallowing it up, bringing it in. Now we have a vacuole. Now, the vacuole I mentioned earlier is like a storage location in the cell. The cell has no purpose for a bacterium. And so what it's going to do is it's actually going to bring in a lysosome. The lysosome is going to dump its enzymes into the vacuole, degrade the bacterium, and get the pieces from it. In this case, we are taking in something that is very large. Now, bacterium is small, right? But compared to a cell, it is a cell eating another cell. So that's a fairly large particle. That is called phagocytosis. Pinocytosis is where the cell takes in basically just water. Think of this as the cell drinking. It needs some water, and so it is going to take in a plasma membrane, make a water balloon basically from plasma membrane, bring it in the cell, and then once it's in the cell, pop that balloon and let that water out. Both of these are what we say are non-selective. They just close, and whatever is in there when they come in gets inside of the cell. Receptor-mediated endocytosis is more specific. Here we have one of those receptors. A receptor is a protein that sits on the plasma membrane waiting for something very, very specific to come in and stick to it. And different receptors behave differently when their specific thing sticks to it. That specific thing is a term. It is called a ligand. The ligand is the key to the lock, basically. And when that key sticks in the lock, the receptor kicks into action. In this case, the receptor causes the cell to carry out endocytosis and bring whatever that ligand is into the cell. Usually we do this as a signaling mechanism. Unfortunately, and this is very timely right now, viruses have hijacked this. they learn to mimic the ligand. And so they come in and they bind to a receptor. The receptor thinks it has captured whatever it wants to capture and takes it in. But when in reality, what it's doing is it's bringing the virus into the cell. And then once it's in, the virus breaks out and all heck breaks loose. The opposite of endocytosis is exocytosis. So exocytosis is easy to remember. Exo, think exit. Endo is a prefix that we're going to use a lot. Endo always means in or internal. So endocytosis is bringing in, exocytosis is sending out. In this case, we have a plasma membrane bubble, but in this case, we've packaged it up inside of the cell. And here, this is called a vesicle. The vesicle is that plasma membrane bubble. Most likely, this came from the Golgi apparatus. The Golgi apparatus will package things inside vesicles and send them off. And if they float out to the edge of the cell, this is plasma membrane, that's plasma membrane, and when they touch, they will fuse together. So here we can see that that plasma membrane is now continuous. It comes along, now it comes down, around, and then continues on the rest of the cell. And if you open that up, you're going to dump these contents out. Sometimes that may be waste products that the cell has and just needs to get rid of, like trash. In other situations, they may be signaling molecules. The cell wants to talk to the cell next to it. So it's going to put out these molecules. This is going to float over, bind to a receptor on the other cell, causing endocytosis on the second cell. Now, before we talked about osmosis. And osmosis is, like I said, a very important concept. If any of you work in healthcare, you've probably injected things into a patient, whether it be medication or just fluids, if someone is dehydrated. If we're injecting large amounts of something, and large amounts could be a couple milliliters, with like a syringe or a vaccine or something, we need to be very aware of what we call the osmolarity or tonicity of that solution. Because remember, we learned about osmosis. Things in osmosis, water is going to move through a membrane to where the concentration of the solute is high. You've probably seen or have at least heard or think about when you put a whole bunch of salt on something. If you've ever had a leech stuck to your foot, or you've put salt on a slug or something like that, you've seen where it causes them to shrivel up and die. It's sucking the water out of them using osmosis. The water is going from inside the leech, where there is little salt, to the outside of the leech, where there is a lot of salt because you poured it on there. The same thing can happen with our cells in our body. Now, this example is specifically for red blood cells. So we're going to give someone fluids IV. And we need to make sure that the concentration of the stuff in that IV fluid is the same as the concentration in the plasma and inside the blood cells in that patient. So osmolarity and tonicity refers to the concentration of all the stuff combined. So it is not just the sugar concentration, it's not just the salt concentration, it is the concentration of all the stuff. Now, if we take a red blood cell and we put it into a solution that has the same osmolarity or the same tonicity, we say those solutions are isotonic. Iso means same. They have the same tonicity inside the red blood cell as outside of the red blood cell. And so no osmosis is going to happen. It's the same concentration of stuff inside and outside, so water is just happy where it is. That is isotonic. If we put it into a solution that is hypertonic, the cell is going to shrivel up. That's on the left here. Now a hypertonic solution, hyper is high, right? Hyperthermia or something like that. Hypertonic is a high concentration. Imagine injecting a very concentrated salt solution into a patient. That concentrated salt solution is going to suck the water out of the red blood cells into the plasma. And so the cells are going to shrivel up. If they shrivel up too much or they stay shriveled up for too long, they're going to die. And that's not good. The opposite of this is a hypotonic solution. So imagine injecting just pure water into a patient. In that case, we have low concentration of stuff in the plasma and a normal concentration of stuff inside the cell. And so the water is going to move towards the high concentration and go into the red blood cells, causing them to swell up. Again, if they swell up for too long, they will die. In this case, though, if they swell up too big, they will actually burst. And so we can see that happening here. And so, everything we inject into a person has to be isotonic. If it is not, we're going to have big, big problems. Does everyone understand this slide? This is a slide that the department really, really likes in terms of exams. Okay, thank you. So here's a review question for you. Water moving through a selectively permeable membrane.
Osmosis.
Yes. Selectively permeable membrane, like I said, will never ever be used in any context other than osmosis. Does someone want to describe to me or explain filtration? Anyone feel confident enough to explain filtration? Does anyone feel confident enough to explain diffusion?
Is filtration just like literally like a filter on a pump?
Yeah, so like she said, it's like a filter with a pump, and the pump is applying pressure, right? We put pressure in one side of the filter and we force things to go through. But some things stay behind. So usually small things will get through, big things stay behind. Good.
The big things you said were cells, right?
So if we're talking about filtration in terms of blood, yeah, it's going to be cells, and we didn't learn about them, but there are also some large proteins in our blood plasma that wouldn't be able to get through.
Okay, but for just so I guess for this particular one, just understand that the pressure is meant to push through the small things and not the larger ones.
Right. Right. Okay. Right. So right now we're just on the concept of filtration rather than examples.
Okay.
What about diffusion?
The diffusion is when you have unequal levels that's divided by the biolayer, and through time it will get to where it comes to equilibrium.
Exactly. Right. So diffusion, in its most basic sense, where if you just go into a physics class, is the movement of things from high concentration to low concentration. And so like he explained, for us though, we're talking about in biology. And so this is a cell. So we have the cell membrane. Maybe we have a high concentration outside and a low concentration inside. And so it's going to want to diffuse across the plasma membrane into the cell. It can also work the other way. If you have a high concentration inside, it can diffuse out.
Okay. I'm a little confused. Is that the example where you have the sugar trying to I guess even out the other side?
So the fusion was...
Like it was
that one.
Okay.
I think the slide you might have been thinking about were you thinking about that one?
Oh, okay, that was osmosis.
That's osmosis. Yeah, so
okay.
in diffusion, the sugar moves to equal out the concentration. In osmosis, the sugar can't get through the membrane, and so the water moves to equal out the sugar concentration.
Okay, water in osmosis.
Yeah.
Sugar and
Yeah. So in osmosis, only water will move.
only water. Okay, got it. So what am I doing now?
The next thing we're going to talk about is the process of making proteins. So this is protein synthesis. We talked earlier about an organelle that makes proteins. What organelle makes proteins? Starts with an R.
Like ribosomes?
Yes, ribosomes make proteins. And where are most of the proteins made?
Mainly the rough ER.
Yes. They're mainly made in or on the rough ER by the ribosomes. Now, protein synthesis is going to be a multi-step process. We know that the DNA is in the nucleus, and we know that the DNA is the instructions. The DNA contains our genes, which are our instructions. So this is DNA. Last week we talked about nucleotides, nucleic acids, and we saw that the different nucleotides, the A, T, C, and G, that make up DNA, and we saw that they make polymers, right? And then I think we talked about how you can take two strands and twist them together to make the double helix that everybody knows about. But the DNA structure is actually more complicated than that. So that's what we're looking at here. So I want to start at the bottom of this figure because that is what you're familiar with. This is the polymer or the string of nucleotides. So on the top we have A, A, T, A, C, A, G, C, G, G, T, T. That is the code for us. And then it pairs up when we get to two strands. We get the double helix, but notice we then take that double helix and we're going to take that string basically and we're going to wrap it around some beach balls. Those beach balls are called histones. The histone is the ball itself that we're wrapping it around, and that's actually a protein. If we look at DNA wrapped around a histone, that mixture of things is called a nucleosome. A nucleosome is DNA wrapped around a histone. We then take those nucleosomes, they look like beads on a string. There's little pieces of loose DNA in between the beach balls. And then we kind of wrap that and make that into a coil. And once we have that coil, that is called chromatin. Chromatin is the storage form of DNA. It's packed in very, very, very tightly. We can't access it very well. Think of it like taking a file on your computer and putting it into a zip folder. You can't just access those files. If you want to access those files, you have to unzip it, right? So if we want to read the code, we have to undo this chromatin so that we get down to this unzipped level down here. Now that chromatin is a material like wood or metal. But you take material and you make something out of it. So we have a wood board or we have a metal beam, right? Here we have a chromosome that is made out of chromatin. So instead of having a board that is made of wood, we have a chromosome that is made of chromatin. But that chromosome is really, at its most basic level, one gigantic DNA molecule wrapped around a whole bunch of beads. And that is a chromosome. And then in each of our cells, we have 46 of these chromosomes. 23 from our mother, 23 from our father. So So if we zoom in on our nucleotides, I mentioned last week that hydrogen bonds were going to be very, very important in DNA. And hydrogen bonds, remember, are bonds formed by the sharing of a hydrogen atom. In the top right here, actually, let's go to the bottom. This is one nucleotide. A nucleotide, the A, T, C, or G, has 3 parts to it. There is a sugar in the middle. It doesn't do a whole lot. There is a phosphate, which acts kind of as the glue to hold one nucleotide to the next one in the string. And then there is this nitrogenous base. Now that is not a term I need you to know, but what I do need you to know is that we have 4 different nucleotides in DNA. The only difference between them is the chemical structure of this nitrogenous base. And if we look at the 4 nucleotides in the top right and look at the nitrogenous base in each, they are different from each other. And the reason we get a double helix is made of 2 strands of DNA that comes together to form a double helix. And those 2 strands are held together by hydrogen bonds. The nitrogenous base on one nucleotide forms hydrogen bonds, that's these dashed lines here, with another nucleotide. Here we see a thymine, a T, hydrogen bound to an A, an adenine, and a guanine, a G, hydrogen bound to a cytosine. Now, they will always be in those pairs. A's and T's, C's and G's. You will never ever see an A with a G or a C. And that's because A's and T's, you can see, form 2 hydrogen bonds, and G's and C's form 3 hydrogen bonds. That's why they are not compatible with each other. And so in that double helix, right next to each other, you have to have C's and G's and A's and T's. That is what we mean when we say they are complementary to each other. They have to be lined up with each other. If they're not lined up, they won't form a double helix. So, we said DNA has our instructions. And what the instructions are instructions to make proteins. Proteins make everything else. Proteins will make carbohydrates. Proteins will make carbohydrate. Okay, is that what I just said? Proteins will make carbohydrates. Proteins will make lipids. The DNA does not code, it does not have instructions to make lipids or carbohydrates. It makes proteins, and the proteins make everything else. And so we need to focus on how do we make a protein. And this is a 2-step process. First we have transcription, then we have translation. So transcription is the first step. In this case, we are going to go into the nucleus, because that's where the DNA is. Remember, the DNA does not leave the nucleus. Think of if you're building a house, you're going to have a master copy of your blueprints. You are not going to take that master copy out to the work site where it can get rained on and destroyed. You're going to make a copy of them. So that is transcription. Transcription is making a temporary kind of disposable copy of the instructions. And that disposable copy is RNA, specifically messenger RNA or mRNA. So mRNA is made in the process of transcription in the nucleus. The mRNA then leaves the nucleus, comes out here into the cytoplasm, into the cytosol, and we undergo the second process called translation. Translation is going to use that temporary copy, the mRNA, the instructions, to make the protein. So DNA and RNA are both nucleic acids. They're very, very similar. The only difference is primarily that DNA uses DNA uses T's and RNA uses U's. They're basically the same. But when we go from RNA to protein, we're going from nucleic acid to protein. Fundamentally different types of molecules. So that's going to be a more complicated process, and we'll see that. You've heard about genes. Genes are just the sequence of nucleotides. That is all they are. If you look at the sequence of nucleotides, that is your genes. When you hear about DNA sequencing, all that is doing is giving a readout saying that this DNA strand is ATGCCGCAATCTGTT and so on. That's all the DNA sequence is. But from that DNA, from that sequence, we have developed the genetic code. And when I say we, I mean biology has developed the genetic code. Every 3 nucleotides codes for 1 amino acid. And so we see that here. So we've taken our DNA and we've turned it into RNA through transcription. So all we really did is notice that this strand is identical to this strand, the bottom strand on the DNA, except we replaced the T's with U's. But other than that, it's identical. Now, what we do is we look at 3 nucleotides at a time. 3, 3, 3, 3. And each combination codes for a different amino acid. So AUG codes for methionine. CCG codes for proline. Down at this end, UGU codes for cysteine, and so on. And so that is how we read this. And so the way I think of it is if transcription and translation refer to, or words that we use outside of biology also. If you were going to transcribe something, a lot of times it's usually going to be taking a video, taking spoken language, and putting it into written language. But in most cases, it's going to be English in both cases. But if we're going to translate something, we're going to take it from one language to another language. And that is a very specialized skill. That is a complicated process, right? Because to do that, you have to understand both languages. To transcribe something, you only have to know one language. Someone who only knows English can translate this video, or not translate, transcribe this video that we're having right now. But it takes a specialized person to translate it into another language like Spanish or French. So this is transcription. Remember, transcription is taking DNA and making messenger RNA or mRNA. Now this is a complicated process, and you do not have to know how this process works. You have to know 2 things. You have to know that transcription is the process of taking DNA and making messenger RNA. And you have to know that this process is carried out by an enzyme, a protein called RNA polymerase. There's 2 parts to this word polymerase. An ACE just tells you this is an enzyme that's going to do something. And this is a polymer ACE. A polymer is a string of molecules. We're making a string of nucleic acids. So we're making a polymer of nucleic acids. And this polymer that we're making is RNA. So it's RNA polymerase. So we carry out transcription, which I said was complicated, and then we get to translation, which is even more complicated. Here, we need to read the mRNA, 3 nucleotides at a time, figure out what amino acid it's coding for, bring that amino acid in, and add it to the string on the protein that we are making. This is really complicated. I know I've said that, but this is a complicated process. remember, this is carried out by ribosomes. Ribosomes make proteins. That is what they do. They make proteins by carrying out translation. Ribosomes move along the RNA, reading it as it goes. And it reads the RNA, and it calls out saying, I need a methionine, I need a serine, I need a proline. And when it calls out for a particular amino acid, something called a transfer RNA or a tRNA is going to bring that amino acid in. And so we see this here. The ribosome has read AUG. And AUG means we need a methionine, an MET. So the ribosome calls out, bring me a methionine. The tRNA brings in the methionine and it puts it in place. The amino acid, or the ribosome, then reads the next 3 nucleotides. And it says UUU. That means I need a phenylalanine. Bring me a phenylalanine. And so a different tRNA brings in the phenylalanine. Now the methionine and the phenylalanine are next to each other, and so they link together. So in the bottom, now we see the methionine and the phenylalanine are linked together. Now we're bringing in the 3rd amino acid, an arginine. So that arginine will get linked to phenylalanine. As we go, the ribosome moves from left to right here on this mRNA strand, bringing in one tRNA at a time, linking or adding one more amino acid, and then the tRNA that already did its job is going to get left behind and it's just going to float away. There are some very, very nice animations of this on YouTube. My suggestion is go on YouTube, find a simple little animation. It'll really help you see what's going on here. I would do it here, but it wouldn't show up very well on the video. So here's a review question for you. Oh, you're not going to know the answer to that. Let me back up. On that tRNA is something called an anticodon. The anticodon is the complementary nucleic acids for the nucleic acids in the 3 nucleotide code. A 3 nucleotide code is called a codon. A codon is 3 nucleotides. So here's one codon, AUG. The next codon is UUU. The next one is CGA. The anticodon is the complementary version of that. So remember, A's stick to T's. But we're dealing with RNA, which has U's instead of T's. And so the complementary nucleotide here to an A is a U. Then we have a U, so it's an A. Then we have a G, so it's a C. Down here we have UUU, so we have AAA. Then we have CGA, and so we have GCU. That is the anticodon. It causes it to stick there. When things are complementary, they like to stick, which causes the amino acids to be next to each other, giving them time to link together. So we just talked about that. That is the tRNA. The tRNA has the anticodon. Questions about protein synthesis, transcription and translation. Anybody? Okay. So do we want to take a break? No, we're almost done. So we'll just keep on going. So the last thing we have to talk about today is mitosis. Mitosis is cell division. There are actually 2 types of cell division. There is mitosis and there is meiosis. Meiosis is used in the generation of sex cells, like eggs and sperm. We are not going to learn about that. That is included in the reproductive system in 2086. We need to learn about mitosis, though, which is very basic cell division. You do need to remember, though, what the difference is between mitosis and meiosis. Mitosis makes two identical copies of the one cell we started with. So we start with one cell, and we make two identical copies. In meiosis, we start with one cell, and basically all we do is we cut it into two halves. And each of the two new cells ends up with half the DNA. In mitosis, what we actually do is before we split, we make a copy of all of our DNA, and so after we split, both cells have a full set of DNA. I don't have a good way of remembering the terms mitosis and meiosis, which one is which. I remember it because if you stub your toe and you injure your toe, it is going to heal itself using mitosis. And that is a healing process. That is not the process of making sperm or egg. And so if I stub my toe, it's going to heal itself using mitosis. Mitosis is just a part of what we call the cell cycle. Cells have a life cycle. They go around and around and around. And so on this cell cycle, mitosis is actually just this one little wedge labeled with an M. There are two major parts of the cell cycle, or two major times when the cell is doing something. Mitosis, which is the M, and S phase, which stands for synthesis phase. In synthesis phase, this is where we are making a copy. We're duplicating our DNA so that we can then divide during M phase or mitosis into two new cells. Between M and S, we have G1. The G stands for gap. This is just when the cell is minding its own business. It's just doing its own thing. Whatever that cell was born to do, it's doing it. If it was born to be a liver cell, it's being a liver cell. If it's born to be a skin cell, it's being a skin cell. But then, eventually, it reaches S phase and starts preparing to divide. And so it makes a copy of its DNA in S phase. Once it finishes, then, it goes back to being a liver cell, being a skin cell, until it is ready to divide during mitosis. It then divides, and we have two new cells that are now in gap 1 or G1. And those two new cells will go through to make 4 more. And they will go through, and then we'll have 8 and 16 and 32 and 64. This slide I'm going to skip. I didn't want to just delete it because if I delete slides without warning, people get mad. But I'm skipping this slide. So this is mitosis. There are 5 phases of mitosis, and then a 6th related phase. And so, I'm going to have to admit, apparently when I practiced this lecture, I did not look at this diagram close enough. What I want you to do is I want you to cross out prometaphase. You do not need to know prometaphase. That is not something most people even talk about. So just cross out this prometaphase part of the table. You need to know the 4 phases of mitosis: prophase, metaphase, anaphase, and telophase. I remember the order because I remember PMAT, P-M-A-T, prophase, metaphase, anaphase, telophase. You also need to know what is happening in each of those 4 phases. So remember, these are phases of mitosis. In mitosis, we are trying to take one cell, divide it into 2. And when it does that, we need to take the DNA that we have already made copies of and organized them so that one copy of each chromosome goes over here and one copy goes over here. If both copies go over here, this cell is not going to get any copies, and that's not good. We need to make sure that one copy of each chromosome goes to each cell. So we start in prophase. These chromosomes are DNA, so they're located in the nucleus. I hinted a while ago that the only time DNA leaves the nucleus is during mitosis, during cell division. And the only reason it can actually leave is because the nucleus dissolves. That membrane, the nuclear membrane, just goes away. And so the DNA inside can then just float wherever it needs to go. So in prophase, a few things start to happen. We go from a DNA in the nucleus that's just diffuse, meaning it's just a cloud of DNA. We can't make out and say that's one chromosome, that's another, and that's another. But then they actually, they condense. And they turn into this X shape that you're seeing in this diagram. And that is actually what they look like. It's this X shape. And you can say that's one chromosome, that's one, and that's another one. So in prophase, we start to see the X's. The nuclear envelope, the nuclear membrane starts to break down. We also get movement of what are called centrosomes. Centrosomes are another organelle that we didn't talk about earlier. Centrosomes, their primary function is to help pull the DNA apart during mitosis. And so we're going to take this cell and we're going to pull the DNA half this way and half that way. And so the centrosome is going to be the anchor. We're going to have an anchor on each side. And then from each anchor, we're going to throw a line out, a rope, attach the rope to the DNA so that we can pull the DNA to the two sides. So the anchor, one on each side, is the centrosome. And during prophase, they start to move opposite of each other. Normally, they're right next to each other and they're kind of off in a corner of the cell. But in prophase, they start to move opposite each other. And we start to get what we call spindle fibers coming off of them. They're like little fingers coming off the centrosome. So imagine my hands are the centrosomes. My fingers are the spindle fibers. The spindle fibers are going to reach out, grab the chromosomes, and then pull them apart. But in prophase, they're just starting to form. And so if you look at a diagram, if you look at a model, the key here that you are looking at prophase is that the nucleus is still partially intact. The nucleus is still partially intact. The DNA is just kind of randomly located. They are not lined up and they are not starting to pull apart yet. That is prophase. We go from prophase to metaphase. Meta means middle. And in metaphase, the chromosomes are lined up very neatly along the middle of the cell. This calls the middle of the cell the metaphase plate, which is true, but that's not a term that I need you to know. But notice that the X's, the chromosomes, are in the middle of the cell and the spindle fibers are attached. Every chromosome here has spindle fibers attached to it. Then we move to anaphase. In anaphase, the chromosomes are actively being pulled to either side. And so you can see we've taken the X's and we've literally ripped them apart. So rather than being an X like that, we've ripped it apart and it's going opposite to either side of the cell. That is anaphase. In anaphase, they're actively moving to either side. Then we get to telophase. Telophase is the final official phase of mitosis. In telophase, we start to reform nuclei. But in this, now we have two of them, because we're going to divide into two cells, and the spindles start to fall apart. The final stage here, which is technically not part of mitosis, is called cytokinesis. Cytokinesis Cyto means cell. Kinesis means movement. And so we are going to take the two cells and we're going to move them apart from each other. We're going to pinch them and then pull them apart. And so you can see this cell in telophase is pinching in the middle, and it's going to break apart into two new cells. On the bottom here, you are looking at fluorescent images of cells. And I've always really liked this. I spent a lot of time in a microscope room doing just this. To me, this is really cool looking. So what you're looking at, the blue here is a stain called DAPI, which you don't need to remember, but it stains DNA. The DNA is the blue that you're looking at. The green is staining the spindle fibers. And so here in prophase, notice that the DNA is kind of still in this blue cloud. And we only are very little bit starting to see spindle fibers on each side. If we jump over to metaphase though, the blue is lined up very, very nicely, right in a line down the middle, and we have these basically little stars on either side connected to them. And then we are going to, into anaphase, we're going to pull them apart. I wish the picture they picked doesn't show this very well. Ignore the green in the middle, actually. This green that's on the here in the metaphase should be out here on the outside also. And then in telophase, you can't really, it doesn't look all that. You can't really tell what's going on. But in order to undergo mitosis, we said we had to make a copy of our DNA. Now again, this is a complicated process. We're going to make it very, very simple though. It's making a copy of DNA is called DNA replication, and it is done by an enzyme called DNA polymerase. RNA polymerase took DNA and made RNA. DNA polymerase takes DNA and makes another DNA copy. That is all you need to get from this slide. So what molecule makes a strand of DNA? A replicase, a polymerase, or a helicase? Yeah, so it's polymerase. One, we just talked about it, and we didn't talk about replicases or helicases. So that is the end. So that is the end of our cell lecture. Are there any questions about the lecture? Then I'll ask about last minute questions for the exam. But are there questions about this lecture?
It's sort of not. I mean, it's like lecture, but anyway, let me just ask.
Go ahead.
So, when people get I don't know what it's called, but I knew somebody who had a replication of or something to do with their bones, and I guess the brother gave DNA to the other brother, right?
So
and then later on, it rejected the DNA, like it accepted it, but then do you know like? What
yeah. So I think you're talking about a bone marrow transplant.
Yes, a bone marrow transplant.
So that is more about blood cells than it's not about DNA. And so what happened was, I don't remember if it was your brother, the person that received the transplant had a problem with their DNA. They had cancer. Cancer is a problem with the DNA. And so their blood cells weren't behaving properly. And so what they did is they gave them a drug that killed their blood cells. And then they took the blood cells from the donor, put them into the recipient, so that the recipient had functional blood cells. And it sounds like initially it worked. The problem is apparently the immune system from the recipient realized these aren't my cells. I need to kill the donor cells. And so the donor cells all got killed, and it was a failed donation, it sounds like.
It it lasted for a while, then it shut it down.
Yeah.
Can I ask you what makes you a match? Like when people do that, what makes you a match in the first place?
So now you're getting out of my comfort area. So to be honest, Wikipedia could probably give you a better answer than I can.
Okay, I didn't know if it has. Is it DNA?
No, so I could tell you all about the cancer and stuff like that, but I can't tell you about the clinical transplant.
Blood matches or whatever that is. Okay, I was just curious. Okay.
Any other questions? Okay. Questions about the exam, or remember it's on the first 3 lectures. And so if you have any questions about the first 3 lectures, we can consider this a review time. If there's something you want to talk about, let's talk about it.
Oh Lord, I wish I would have wrote questions down.
If you don't have them now, you can always send them to me later. But if you want to talk about it in person right now, we can do that.
Okay. Yeah, I'll write them down. I want to go back through. So this lecture is included?
Yes. The exam is lectures 1, 2, and 3. And if you ever question that, remember that information's in the syllabus.
Okay. And then just to refresh, the other exam is separate, right? Completely separate.
The other exam, meaning the lab exam?
Yes.
Yeah. So the lab exam is completely separate. You have a few weeks left for that.
Okay.
Yeah. So right now, by Monday, you have to take lecture exam 1, which is 50 multiple choice questions. Not a huge deal. 3 lectures.
Why do you say so, the professor? Okay.
I have a question. So, I know that you said for chapter 2, there's not going to be. Did you say that there wasn't a lab for chapter 2?
Correct. For chapter, well, so that confuses people. We don't necessarily go in that the lecture number doesn't necessarily match up with chapters. Eventually, we get off a little bit, and so go by topic. But what you're talking about is the chemistry. Lab 2, the very last portion of it is a very, very, very small chemistry portion, but it's an experiment based, and so you don't have to do it. So you essentially functionally have no chemistry lab whatsoever. Lab 2 is mostly on cells.
Okay.
Yeah. We had a chat question asking if the test questions are similar in difficulty to the homework questions. Yes. Like I said earlier, about 20 of the 50 are directly from the homework, and the other questions are similar in difficulty. They may be worded a little bit differently because they're not from our textbook. Some of them I wrote, some of them are from other textbooks, but I picked them. And so I think they're good questions. I know how difficult they are, or at least how difficult I feel they are, and I think they're the same difficulty as the homework.
Okay. So, would you say that the learning outcomes would be just like I think I'm just a little confused on what exactly the learning outcomes are for, besides the final exam, or is that pretty much what they're for?
So, the best way, in my mind, the biggest function of the learning outcomes is to tell me what to teach you. To me, they're more important to me than you.
Gotcha.
Everything in the learning outcomes is in the slides. I went through, I don't know, there's a couple hundred of them. I went through every single thing in the learning objectives are in the slides, but you can use that as kind of a test for yourself.
Okay.
Just go through the first three lectures with the study or learning objectives and just say, yes, I can do that. Yes, I can do that. No, I can't do that. I better go study that.
Okay, that makes sense. Thank you.
You're welcome. Other question, yes, asking whether the material is spread out equally between the 3. They are spread as equally as possible. Yeah, so it literally is as close as possible. 50 is not divisible by 3, and so it's not perfect.
Is this test worth more or less than the next test?
No,
Is it
so
the first one, or they're all equal?
so we have 4 lecture exams. They are all equal. We drop one of them.
Oh, right.
Right. The lab exams are worth. I don't have the syllabus in front. The lab exams are worth a little bit less, I believe, and the final exam is worth considerably more.
And are we going to be able to like go over this? Like, if you have questions, is it going to be as a class or just hit you up on the?
So that's a good question. And so actually, the only way you will be able to see which questions you got right or wrong will be to set up a individual appointment with me to get online and look at them together. You won't be able to see them. You won't be able to. I'm trying to protect the exam for future, and so I can't just give it out.
But you can tell us so we can like try and study and get that correct.
If we meet together. However, a lot of times that is not, probably, to be honest, not a great use of your time, because when you take the exam, and I mean I've taken plenty of exams myself, you know, I knew this, I didn't know that, and wondering whether you got this one particular question right or wrong, you're not going to see that question again. I write the lecture exams, someone else wrote the final exam, and so going back over the questions I wrote are not really going to help you for the final exam. And so we can do that. I'm happy to get on and go through it with you, but it's not the fantastic study opportunity that you might think it would be.
Okay, what about just like saying, well, this is like out of 1, 2, and 3, you really need to go back and review this particular subject, I guess.
Yeah, so I think I would worry less about, I didn't know this particular detail, but if you take the exam and you say, wow, all the questions that had to do with this subject, I knew I struggled with. Whether I got them right or wrong, I know I struggled with them.
Yeah.
That should be your cue that I need to go back and restudy that.
Yeah, I probably just ask afterwards because like now I can tell what I'm understanding the most, and yeah, I understand because you kind of know already what's what.
Yeah.
Okay.
Other questions? Okay. So remember, there's basically no makeups, and so take your time studying. Don't take it tomorrow or something like that. But don't wait until the last minute. Don't wait until the last minute. And remember, if this is the first exam, you don't know what this exam is going to look like, right? And so if you go in and you bomb it, but you tried, take it as a learning opportunity. Say, I'm going to drop this one. Now I know what these exams look like. I'm going to do better on the rest. It's not an end of the world type of a situation.
So I have one quick question.
Yeah.
If we do have a question about the about material for the exam, do you prefer us to message you in Canvas or like post a discussion on Canvas?
If your question is specifically to me, I would say send me a message. If you're interested in getting feedback from classmates also, post a discussion. With the discussion, I don't think I immediately get an email saying it was posted.
Gotcha.
When I log in, I'll see it. There will be a little pop-up saying it's there. But it may take a little bit longer for me to see it.
Okay, thank you.
Anything else?
No, thank you.
Well, I hope you have a good week. Good luck on your exam. I hope you can all go fishing right now. And see you next week. If you have any problems, just let me know. Just send me a message. Find some way to let me know. I'll do what I can. But
All right.
have a good night.